Just give everyone a minute or two to file in. Okay, well, I want us to get started on time today. So I just want to welcome everyone. My name is Jennifer Wood, and as past president, I have the privilege to introduce today's Society for the Study of Reproduction webinar, sponsored by the Opening Gates Global Reproductive Science Research Capacity Building Grant from the Gates Foundation. This grant was awarded to SSR and the Frontiers in Reproduction to advance research in contraception with a focus on improving human health by developing contraceptive technologies, particularly in low and middle income countries. And this webinar represents an educational component of the grant. So for today, our speaker is Dr. Stephanie Page, who is the head of the Division of Metabolism, Endocrinology, and Nutrition at the University of Washington, where she holds the Robert B. McMillan Professorship in Lipid Research and is the founding co-director of the UW Medicine Diabetes Institute. Pertinent to today's webinar, Dr. Page runs a large clinical research program focused on male reproduction and the development of effective and reversible male contraceptives. In addition, her group investigates the impact of testosterone on male aging, metabolic health, and disease risk. She has an overall interest in disease prevention, including strategies to minimize unplanned pregnancy and metabolic disease across the globe. Dr. Page has a strong commitment to diversifying our healthcare and research communities and has been recognized by the University of Washington as a gender equity trailblazer and as a recipient of the William J. Bremer Mentorship Award. She earned her bachelor's degree from Stanford University and her MD and PhD in immunology from the University of Washington. She continued her training at UW, including residency in internal medicine and fellowship in endocrinology before joining the faculty in 2003, where she's been very prolific with over 120 peer-reviewed articles published to date. Today, she's going to share her journey to develop a novel and reversible male contraceptive in her presentation titled Male Contraceptive Development from Bench to Bedroom. Dr. Page, thank you so much for joining us today. And I just want to note that you can type questions in the Q&A, which Dr. Page will answer at the end of her presentation. So take it away, Dr. Page.
Thank you, Dr. Wood. I'm just going to share my screen here. Welcome, everybody. All right. I guess I'll hear from Jennifer if people can't see my slides.
You're all good.
Great. So thanks for joining this morning. I really hope by the end of this that those of you who aren't already convinced will understand our need for increased contraceptive options and really the barriers and non-barriers to male engagement and contraception. I hope to impart upon you the basics of the biology of novel male hormonal and non-hormonal contraceptives. And lastly, try and address questions which are often raised about predicting the timeline and the potential barriers for introduction of these methods to the marketplace. So there's my disclosures. So I don't think I need to convince anyone who joined this webinar that we really have an epidemic of unplanned pregnancy, and our best tool for preventing unplanned pregnancy is contraception. Despite the really important innovations in female methods, we still have a relatively stagnant rate of unplanned pregnancy throughout the globe, ranging between 40 and 50% of all pregnancies. That's not to say these pregnancies are not wanted, but they are unplanned, and we know the importance of family planning in mitigating the health risks associated with pregnancy and with raising children, as well as the economic risks, not to mention the health and economic risks of abortion. So really, we need a game changer to make a huge impact on this incredible global burden. And really, I'd like you to be convinced, at least at the end, that we have an untapped resource in engaging our male partners. So men actually are engaged in contraception across the United States, and it's a little bit lower globally, but it depends on the setting. Men are participants in contraception and account for about 25 to 30% of global contraception. This is mostly with condoms. About half of that is male condom use. Vasectomy has differentiating uptake across the globe, but is an important but non-reversible contraceptive. And then withdrawal, while it's better than nothing, is considered a male method, less effective. So the last innovation in male contraception, reversible male contraception, occurred over 400 years ago. So I think that we can all agree that we are ripe for something new. Condoms are very important in sexual health, not only for pregnancy prevention, but for STD prevention. But they do have both a high discontinuation rate and a pretty high failure rate at 13% in real use. So they're good, but they're not great as contraceptives. Vasectomy, on the other hand, is exceedingly effective, but it's invasive, costly, and really we think of it as an irreversible method, although there can be vasectomy reversals. That is an even more expensive endeavor. And there are some potential side effects for vasectomy. And then, as I mentioned, withdrawal and periodic abstinence, while better than nothing, really do have a high failure rate. So we'd like to really have more in this menu of contraceptives for men. So, before I get into the types of contraceptives that are in development, I think it's important to set the stage of, who might use a male contraceptive. Are men willing to do this? Are women willing to trust their partner? So I want to go through some of the data about that first. So there are many men who want to participate in family planning, and more survey data suggesting men are interested in controlling their own fertility in some settings. As you all are well aware, many women are unable to use female birth control methods, particularly hormonal methods, because of side effects or contraindications with other medications or health conditions that they have. And there are many settings where couples wish to use two methods. While this can be cost prohibitive in many low and middle income countries, we know more contraception is better. So if there's an opportunity for two methods or a need, that could be useful as well. So, what about survey data? Would men use contraceptives and would their partners trust them to do so? So, for a long time, we relied upon some very good survey data from men that was across multiple countries and included both men and women. And these data from the early 90s and 2000s supported the notion that many men are interested in contraception and women in committed relationships across the globe are willing to trust their male partners to use male birth control. But these are difficult, right? We're asking people hypotheticals. How much does this translate into the real world environment? And we still don't know the answer to that. We do know from the clinical trials, some of which I'll talk about today, that men who engage in our clinical trials are exceedingly happy with the methods in general, would use them if they were available. And in couple studies that I'll show you, women involved in those studies are also very happy with the method. But of course, that's a very biased sample, right? Those are people that are willing to enroll in a contraceptive trial. Certainly, engagement and uptake would depend on the side effects, and in our current world, also on just the perception of what the side effects might be. But we know from female birth control methods that as these products are introduced, there's actually usually a difference in uptake than we might have expected, and often more uptake, and that the economic, and as people are continuing to understand the economic costs of large family size in some settings, social norms can change as the technology is advanced. So the Gates Foundation actually supported, together with the Male Contraceptive Initiative, a really important study that was published in 2024, led by Steve Kretschner. And this was a very big survey study. It was actually complicated because it's what's called a discrete choice experiment, where they were trying to not only understand potential uptake, but what barriers are, what people's choices are driven by, and whether introduction of novel male contraceptives would increase uptake or simply replace female methods. This survey was done in 6 countries: the United States, Kenya, Nigeria, the Cote d'Ivoire, uh, Congo, the Republic of Congo, Vietnam, and Bangladesh. There were over 12,000 men that were surveyed and 9,000 partners in most of those countries. The survey was not done in females in the United States. And I think the really important take home is shown here in this panel, which is asking people if a new method was introduced to the market, would they be willing to try it within 12 months of introduction to the market? So this is meant to control for people who say, oh yeah, I'd try it someday, but use in the first 12 months is more correlated with actual uptake in other types of health interventions. And you can see here that in low and middle income countries, the willingness to use a novel method was exceedingly high, lowest in the United States, but nonetheless, overall, people, as methods would continue, would be more willing to try them. So this is higher than Steve ever imagined when they conducted this experiment, this survey, with upwards of 70% of men being willing to try a novel method. Importantly, they did work to try and understand if this would attract new users. And you can see here in the green, this is potentially new users, not people who are switching from female methods or switching from male methods. So potential very large impact here in terms of uptake and new users of contraception who are not currently using contraceptives but are at risk for pregnancy. And then just as importantly, women were willing to trust their partners to use these novel methods. So again, it varied by country, but you can see that in most of these countries, more than 50% of women would trust their partners to use a novel male contraceptive. So this is really exciting data, I think, for the entire field, and again, represents probably an evolving in norms about male sexual health and contraception in general. So we don't have very many methods for men, and we think that that's a big reason that there is less engagement for men in contraception. So what we would like to create is a method of male contraceptives that mimics that of the options that are available for women. So down here, we have methods that are on demand or need to be or are user controlled. As things get longer in terms of their mechanism of action, their failure rates tend to go down with female methods, and there's no reason to think that that wouldn't happen for men. And again, long-acting contraceptives are most effective. But all we have now for men is something on either end of the spectrum: vasectomy, not really reversible, and male condoms. So we would like to fill in this middle area here and provide men with a multitude of reversible options that we are convinced would increase uptake of contraceptives. So again, Steve's work, sponsored by the Gates Foundation, was also very helpful in trying to identify the mode of administration that would be that men are most interested in. And I think this was also a surprising result in that the men in many of the countries surveyed, preferred a topical transdermal gel, which is something I'm going to talk about later in the webinar. In the United States, the preferred method was a pill, but in many countries, it was a transdermal gel. But I think the take home from this slide is that men are interested in user-controlled, self-delivered contraceptives that don't require them to continue to go to the clinic. So how do male contraceptives work? And just a review of male reproductive biology, which not everybody thinks about every day. So spermatogenesis happens here in the testis. There's a whole bunch of steps that go from the development of sperm stem cells to the production of sperm, and a lot of it happens here in the testis. Then the sperm exit the epididymis. Again, there's more maturation of the sperm in the epididymis. And then once the sperm reach the female reproductive tract, they have even more steps to go through to attain functional motility, which is hyperactivity, and then for fertilization. So there's multiple points one could think of in the biology of male reproduction that we could intervene in to develop contraceptives. What I'm going to talk about initially, because it's most these methods, hormonal methods, are furthest along in development. It works very, very early on. So hormonal male contraceptive methods block spermatogenesis in very early sperm development here in the early stages in the seminiferous tubules. So we're often asked, why is it taking so long? What is so challenging about developing male contraceptive methods compared to female methods? And obviously, the important imperative with the initial stages of developing contraceptives was to develop female methods because women, of course, bear the burden of unplanned pregnancy. But there are some other challenges. And then one of them is that it's really a numbers game. So people are sometimes surprised to learn just how many sperm are in an ejaculate. So there's 15 to 200 million sperm per milliliter in a normal male ejaculate. And yet we know it only takes one sperm for fertilization. So a lot of work was done early on as these methods were being developed. And from early studies from the WHO that used testosterone as a contraceptive, we were able to delineate that even at least in a clinical trial setting, if we can get sperm concentrations from that 15 to 200 million per mil to just less than a million sperm per milliliter, that correlates to a contraceptive efficacy, at least in a clinical trial setting, that's comparable to perfect use oral contraceptives. So that's the target we use. We don't need to eliminate all the sperm, which I think is surprising to many people. But if we can get the sperm concentration to this threshold, we know that it's compatible with effective contraception. So how does hormonal contraception work? So it's very similar to how female hormonal contraception works. In men, the pituitary the hypothalamus secretes gonadotropin releasing hormone in a pulsatile faction, which acts on the pituitary, which in men, just like in women, secretes FSH and LH. In men, the LH acts on the Leydig cells which produce testosterone, and FSH acts on the Sertoli cells, and together these are both important for the development of sperm. When we give exogenous testosterone, we block all of these things by taking advantage of natural negative feedback. So this is how the brain and the testicle keep everything in homeostasis in normal healthy men. We give exogenous testosterone, the negative feedback blocks the production of GnRH, LH, and FSH in men. But we're giving the testosterone, so it supports male secondary sexual characteristics like muscle mass, sexual function, and male pattern hair, but it blocks the endogenous production of testosterone. So plenty of testosterone around, but the testicle is no longer making testosterone. And the sperm, it turns out, require that high concentration of testosterone that's usually found in the testicle, about 1,000-fold the concentration of testosterone in the blood. And without that testosterone within the testicle, the sperm do not mature. So early studies done again by WHO and others demonstrated that conceptually this works. So if you give men high doses of testosterone, it turns off all of these signals, and we can get many men to this threshold of less than a million sperm per milliliter in the ejaculate, and we have effective contraception. Importantly, if we add a progestin, just like with female contraceptives or some hormonal female contraceptives, the combination of testosterone and the progestin is more effective than testosterone alone. And that's because the progestin really helps at the level of the brain and the pituitary to block that production of LH and FSH. And so we get better blockade of spermatogenesis when we use a combination of testosterone and a progestin. Okay, so the way these contraceptives work, again, the hormones that we give the men, the androgen in this case, plus the production, block the production of gonadotropins. And that happens very quickly. So on this panel here, you can see the production of LH on the y-axis and days on the x-axis. And when we give these combinations, of testosterone and a progestin, the it happens really fast. So LH is blocked very quickly, and this just shows that we add the progestin, in this case nesterone, to testosterone. That blockade and lowering of gonadotropins happens quickly and is very marked. So what does this mean in terms of sperm production? So on the other panel here, you can see that even though the gonadotropins are reduced very, very quickly, spermatogenesis in men occurs over a process of about 72 days. And since we're blocking very early, the sperm that are already past that point in development when we start giving the hormones continue through. So it's a very specific block in early sperm production. And so this means that hormonal methods really take 8 to 12 weeks to work, because we have to not only block the production of the sperm, but wait for those sperm that are already past that checkpoint to mature. So the on rate and the recovery rate of male hormonal contraceptives is going to be 8 to 12 weeks. And we've seen that in many studies. When we add the progestin, again, it's faster and it's more complete. So modern male hormonal contraceptive regimens include both an androgen and a progestin. So this combination technology has been in the works for a few decades now, and multiple different studies have demonstrated that we can give testosterone or the androgen in all kinds of different ways, intramuscularly, as a transdermal gel, as an implant, and we can combine it with all kinds of different progestins. And overall, we can get about 90 to 95% of men to that threshold, less than 1 million sperm per milliliter, that we know is compatible with effective contraception. So it works in almost all men. All men have a decrement in their sperm production. There are a few outliers who don't reach that threshold, and we still need more work to understand what's different about those individuals. But overall, we can get most men, the vast majority of men, to this threshold concentration. So what about side effects? So we're constantly asked about side effects, and side effects are concerning to both potential users, but also to regulatory bodies who we will need to get these methods approved by in order for them to actually reach the marketplace. So early studies of male contraceptives use high doses of androgens because they didn't include a progestin. And in those studies, we did see androgenic side effects, which included weight gain, acne, increases in hematocrit because testosterone actually drives erythropoiesis, and changes in cholesterol with some changes in libido. With the inclusion of a progestin, we are able to reduce the dosing of the testosterone to be physiologic. So the men who are on modern male hormonal contraceptive regimens or trialing them are having normal concentrations of testosterone in their blood. And this really minimizes those androgenic side effects. Moreover, with use of more progestin-specific progestins, we've also been able to really streamline and minimize side effects. So what we see in these more modern regimens is very minimal weight gain. We do occasionally see acne. The changes in hematocrit have been nullified with the normalization of the testosterone levels, and we don't see any changes in cholesterol. We do a lot of work to try and understand potential changes in libido or mood. And again, I'm happy to report with these more modern methods, those are very minimal. And so the question will always be, is this side effect profile acceptable to users? And really, what are the regulatory agencies going to require in terms of side effect minimization? Because in this case, we are giving the hormones to the person who does not have a direct health benefit from the prevention of pregnancy. That's how the regulatory agents look at it currently. I'll talk at the end about work we're doing to try and change that mindset. But with there being no health benefit for the user, the amount of risk that may be acceptable to regulators might be very low. There are some potential benefits. We know that giving men androgens can improve their bone density, can help with maintenance of lean body mass, and in many instances, improves libido. Okay, so that's sort of the background. as I said, there's been lots of studies that demonstrated that we could get men to these threshold concentrations of sperm we know are compatible with contraception. But really, the proof is in the pudding in terms of preventing pregnancy. And to date, there have been six clinical phase 2 efficacy trials. So, what we first do is do safety studies, then we demonstrate that these products reduce the concentration of sperm to the threshold needed for contraception. And then we do studies in phase 2 and 3, testing these products in couples for the prevention of pregnancy. So there have been six efficacy studies to date of male hormonal contraceptive involving approximately 2,000 couples. And I'm happy to report that well, it's not the largest, the most recent and first phase 2 study of pregnancy prevention of a user-controlled male contraceptive has just been completed, and I'll show you some of that today. And those results are promising. So that's been a major advancement. Again, all of these products have efficacy of 99 to 95% in these clinical trial settings. They're all reversible. The reversibility is very important to emphasize. While it can take 8 to 24 weeks for men to achieve normal sperm concentrations, they are reversible, just like hormonal female methods. It is important to note that we don't have a phase 3 study. So the hope is in the next 3 to 4 years to initiate a phase 3 study of one of these methods, which will be required in order to introduce them to the market. Okay. So this transdermal gel, which some of you may have heard about, it has had a little bit of media activity. So what's new about this compared to the studies that have been done in the past? So this is a gel that men put on themselves. So instead of having to go to the clinic for injections, this is a user-controlled method. We know that giving hormones transdermally, particularly testosterone, provides very stable pharmacokinetics. Men seem to really like using gels for androgen replacement. Testosterone gel is the #1 method of replacing testosterone in men with hypogonadism in the United States, and this gel is built upon that technology. And importantly, Nesterone, which is been developed by the Population Council, is a very progestin-specific progestin, which means that it doesn't really have androgenic or glucocorticoid cross-reactivity. And so we think this is very important for minimizing side effects. And then in terms of potential use across the globe, these gels are relatively heat stable. They don't require refrigeration, and we know that they're stable between about 18 and 38 degrees Celsius. So in terms of, again, user control and dissemination, that's a very important component. What does it look like? So this is the actual gel we used in the study I'm going to talk about. It's about the same, it's very similar to hand sanitizer. It's an alcohol-based gel. The men put one pump in one hand and then one in the other, and they rub it on their shoulders, wash their hands afterwards to prevent any transfer. So we initially did studies to just determine the appropriate dose of Nesterone using suppression of LH, the gonadotropin required for testosterone production in men, to try and figure out what dose of Nesterone would be tolerated in combining it with testosterone. And we settled on this 8 milligram dose that you see here because it gave maximal suppression of LH. And then we did the appropriate sperm suppression study, which I showed you before. Here we're comparing giving men testosterone alone or 2 different doses of the Nesterone. And you can see that the 8 milligram dose of Nesterone was just as effective as the 12 milligram dose, resulting in sperm concentrations below that 1 million per milliliter threshold in about 8 to 12 weeks in men. So that was very encouraging. Really no side effects of concern. So we went on to do a phase 2b study. So this is a sort of a flow diagram of what we do in these studies. So we screen couples. We enrolled 462 couples across the globe with a goal of completing 200 couples who relied on this method, this gel, for 1 year of contraceptive efficacy, relying on it as their only method for contraception. We were able to include sites across the globe. This is the first time that we've included um, uh, male contraceptive study sites in Africa. We had a site in Zimbabwe and a site in Nairobi, so that was very exciting, as well as sites in Chile, North America, and Europe. So the way that we design these studies is the man and the woman enroll. They're both consented because, of course, the woman is taking some risk, significant risk. And then the man begins to apply the gel, and we wait until his sperm concentration reaches that threshold of 1 million per milliliter before they stop using their current method of contraception. They then enter the efficacy phase. They the man is monitored monthly, the female partner every 3 months to make sure that his sperm concentration remains suppressed. And after completion of that efficacy phase, they are monitored for recovery, which is really to ensure that his sperm concentration returns to normal. So the primary endpoint of this study is prevention of pregnancy. We completed it in 2024, and we've just submitted the primary results for publication. So while I can't share that, I can tell you that we are very encouraged from the results of this study. It is important when we interpret those to remember the study design, which is that we did quite a bit of monitoring during the efficacy phase. And when we think about moving this forward into phase 3, we have to design a study appropriately that is more mimicking of real use. But it was very exciting to do. We are hopeful that you'll read the results when they come out in the next year or two. And I can tell you that there's lots of interest in moving this product hopefully forward to phase 3. So as I just mentioned, we did a lot of monitoring of sperm concentrations during the study, and we sort of have been asking the question, would people want to do this themselves in order to feel reassured, particularly the female partner, that that the man's sperm concentration is still suppressed? And so we did a sub-study nested within this trial of using a device that had been adapted from post-vasectomy monitoring devices, and to see if we could, if people at their homes could actually monitor their own sperm concentration. And happy to report that this worked really well. These tests are very similar to the COVID tests that people became quite familiar with when we were doing this study, which happened across COVID. We compared that to what we were seeing in the laboratory. So the point here is that there are devices that could be used in conjunction with visits to the clinic or in replacing visits to the clinic that would help people with sperm monitoring if they felt that they wanted to have reassurance about a man's sperm concentration. Okay, so that's the gel. We also know that men are very interested in pills. They see their female partners take them. Everybody thinks pills are going to be really easy. And so there's a lot of interest in a male pill and has been for many years. So why don't we have a male pill? We have a female birth control pill. What's the holdup here? So it turns out that delivering testosterone orally, in contrast to estrogen, there's really very poor bioavailability. And so early in the 70s, actually, there were modifications made to testosterone, a product called methyl testosterone, which is actually approved, which overcomes this. So methyl testosterone has a long serum half-life, but unfortunately is hepatotoxic. So not recommended for any use at all whatsoever, but of course not for a male contraceptive. In the 70s, it was recognized that if we add long-chain fatty acids to testosterone, we could improve the oral bioavailability. But as you can see from this graph I'm showing you here, even these modified forms of testosterone require twice daily dosing in order to maintain men's testosterone level in that normal range, which is very important for their sexual and for other reproductive health. So We know it's hard to take a pill every day. There's no way we think that a twice daily oral pill would be an effective contraceptive. So that's been the challenge. But we've been working with the NIH on developing some novel androgens, so modified forms of testosterone that have longer half-lives and might be able to be delivered orally. And what's really exciting about these molecules is they're not just testosterone. They actually have been modified so they have both that progestin component as well as the testosterone component in one molecule. So instead of having to give both a progestin and testosterone, we're hoping that we can just give one pill or one injection and get the job done. So there's two compounds we've been working on. One is called dimethandrolone. The other has a super long name, so we just call it 11-beta MNT. These have this long chain fatty acid again to improve their oral bioavailability, and then that's cleaved off naturally in people with naturally occurring esterases to these active molecules, which again bind both the androgen and progestin receptor. So we did some early phase 1 studies that looked good. We learned that these molecules, like many lipophilic molecules, have to be given with food in order to be effective. And then we had the opportunity to do a 1-month study. So I'm just going to walk you through this here, just so you have a little bit of an understanding of how we monitor these hormones. So this is gonadotropins on the top panel and testosterone on the bottom. The participants are admitted to the clinic. They do 24-hour blood sampling to demonstrate, to understand what's going on in that first day. You can see in the first day, they're already starting to see some lowering of LH and testosterone. And if you focus on the green and the blue, which are the highest doses that we used, we then send them home. They take the medication every day. We have them come to the clinic twice a week to understand what's happening, make sure they're doing fine. And you can see that by the third day, they have almost no LH and almost no testosterone. And that when we bring them back to the clinic in 4 weeks, these really showed very nicely this total suppression of testosterone and LH, which we know is compatible with suppression of sperm production. The study's not long enough to have measured sperm production, but we know these hormonal changes, and so should be effective, and so that's very exciting. And we did this with both of the two compounds. So you might ask, oh my goodness, with those really, really low testosterone levels, didn't these men feel terrible? Those are castration levels of testosterone. But remember, these molecules that we're giving them are really potent androgens. So even though they're not in the testis supporting sperm production, they're supporting all the other male sexual characteristics, and these men felt fine. So they did not have hot flashes. They did not have decrements in their libido that were substantial. So we know we were giving them plenty of androgen despite them not being able to make their own testosterone. So that's very exciting for us. And we're hopeful that we'll be able to move this forward into longer dose studies. The other thing that was kind of interesting is because this was a clinical trial, we had to do all this different doses and so forth. The participants actually had to take 4 pills every day. They had to take them with food. It was kind of a pain in the neck, but they were all really, most were very satisfied and would recommend the method. So that was also really exciting to see. And again, we hope to move this forward and ultimately end up with a male pill. We're also similarly developing these as long-acting injections. We'd like men to have something equivalent or similar to long-acting female methods like Depo-Provera, hopefully with a better side effect profile. And we've been working on developing dimethandrolone as a long-acting injection as well. Okay, so that's the hormonal methods. If I had been giving you this talk 3 years ago, while I would talk to you about non-hormonal methods, I would have said there's no non-hormonal method that's made it to clinical trials that's still in development. But I'm really excited to say that that is no longer the case. There are 2 non-hormonal male contraceptive methods that are in clinical trials that I'll go over. So why use non-hormonal methods? I think that we often hear, well, if it's not hormonal, it won't have side effects. I don't think that we really know that actually. There's really no drug that doesn't have any side effects in anyone. But certainly these non-hormonal products should not impact testosterone-dependent functions such as sexual function and body composition. And there is in some circles these days, some negative connotations surrounding hormones. Although I would say the marketplace for testosterone supplementation in men is very strong, too strong probably. And so I'm not sure that those connotations exist for men in the same way they do for women in the current marketplace. Um, while we don't think there will be hormonal side effects, one of the things that has to be addressed in development is any non-reproductive or off-target effects, and that's really challenging in preclinical development. And then, if we're not blocking sperm production with these agents, what is going to be the surrogate marker that we can use before relying on them as contraceptive? So that's sort of the background on non-hormonal methods. Again, it's a very exciting field right now in male contraception. So these methods could impact all kinds of places again in the male reproductive pipeline. I'm going to go through the ones that are in clinical development, but importantly, um, while hormonal methods impact or intervention point is in early spermatogenesis, many of these new methods, non-hormonal methods, are trying to impact things like sperm motility and even the acrosome reaction, things that are further in development in sperm development, which may allow them to have shorter on rates, so not that 8 to 12 weeks on and off rates, because the impact would be later in sperm function. If you think about this in terms of the methods we currently have, we really only have things that are impacting the plumbing, so to speak, or barrier methods with vasectomy and condoms. And again, these non-hormonal methods could potentially impact things further down the sperm development track. In contrast, there are also some non-hormonal methods, including things that interfere with the retinoic acid synthesis pathway that I'll talk about, that also impacts and blocks spermatogenesis. And we would anticipate that those methods, like hormonal methods, will have slower on and off rates just because of their biology and how they work. Okay, so there's a whole bunch of things in development. I'm gonna focus on three different um, uh, modalities. One which impacts sperm motility, a second which is really a reversible vasectomy, and then lastly these retinoic acid biosynthesis inhibitors. And I've chosen these because these are again the furthest along in clinical development and things that we might be seeing reaching the marketplace in the next hopefully 5 to 10 years. So retinoic acid, it turns out, is absolutely critical for spermatogenesis. And we know this from mice because retinoic acid is a vitamin A derivative. And if we deprive mice of vitamin A, they become infertile. And that's sort of one of the proof of concepts of the importance of retinoic acid in spermatogenesis. And so people have been working on this for a while. Dr. John Amory at our institution at the University of Washington has been working on things that interfere with the synthesis of retinoic acid, and a group at Columbia and Minnesota, and now at a company, have been working to develop retinoic acid receptor antagonists, which also interrupt this pathway. And importantly, one of those has now reached phase 1, and that's been recently published by Your Way and the group of collaborators. So that's really exciting. It's an orally delivered antagonist with the hope of blocking spermatogenesis. So that study was recently published. So this is just single doses of this retinoic acid receptor antagonist that was done fasting. And these are just single doses, so we don't know what's going to happen with multiple dosing. But I think one of the exciting things about this study is that they showed that these compounds last in the serum for a considerable amount of time. And so when we think about oral daily dosing and and what we know is that sometimes people forget and so forth. The fact that this compound, if it works as a contraceptive, lasts for a long time in the blood is really exciting. So we're looking forward to more work from your way on these retinoic acid receptor antagonists. Um, so that those would block spermatogenesis. What about vasectomy? So we know vasectomy works. It's great. But we really consider it irreversible. So um, a few different companies, and importantly, Contraline in the United States, because again, they're a little bit further along, have been trying to work on methods that would result in a reversible vasectomy. So by using hydrogels that they inject into the vas deferens, that tubing that connects the testis and the outflow tract. Um, they hope to, the concept here is to inject a hydrogel that either naturally dissolves or to go back in and put in a dissolution if the person wants it to be reversed. So the two-step process has been difficult because of potentially scarring and showing the reversibility. But Contraline has started a phase 1 study in Australia to try and wherein they inject this hydrogel and it naturally dissolves. And so we're really excited to see what happens with this reversible vasectomy device. So again, in phase 1 trials with a different regulatory pathway because it's a device and not a systemic drug. And then lastly, this is preclinical development, but there's been a lot of buzz about on-demand male contraceptives. So the sort of concept here is, what about a Viagra for men that was a contraceptive that they could take a few hours, a few minutes before intercourse that might act as a contraceptive? That would be really exciting. And so the target here is to impact sperm motility. So sperm have to be activated to do this hypermotility piece where they can swim in a very organized and yet fast, if you will, way once they get in the female genital tract. And people have identified proteins, in particular this soluble adenylate cyclase enzyme that are critical for that sperm motility piece. So the idea is that the person could take this inhibitor and that it would interact with the sperm for a short time and inhibit the motility so that ejaculated sperm could never get to the oocyte or to the egg, I'm sorry. So this group from Cornell published some work in mice in 2023 that was very exciting, that showed not only, as you can see in the panel above the graph, that these systemically delivered inhibitors of soluble adenylate cyclase inhibited motility, but also when they gave them to mice and then mated them, that they saw contraceptive efficacy. In other words, no pups for a finite period of time. So reversibility after 2 to 3 hours and good contraceptive efficacy for that window. So this group and others that work in this field are trying to move this forward as a male on-demand contraceptive. So more things to come, I hope, in terms of development of these novel reversible short-acting male contraceptives. Okay, so just lastly, I just wanted to talk about regulatory issues and why don't we have these things in the marketplace. And one of the reasons I think is that traditionally regulators and ethicists to some extent have thought about male contraceptives in the context of the male only. So again, there's no health risk for pregnancy for men that really has been documented. There may be some mental health risks and so forth. There's some data that shows that. And so I think what we really need to do as we continue to evolve as a society and think about family planning as a dyad and wanting to improve the health of all of our population, move towards a concept where we have a dyad risk model. In other words, that we're willing to accept some risk for the man in preventing the life-threatening risk for the female. And this is not a novel concept. We are willing as a society to accept health risks for no benefit of ourselves. And I think the classic example here is that it is permissive to do living donor organ donation, kidney donation. That provides no health benefit for the user, but obviously a great health benefit to the rest of society. And if we think about that, about contraception with that sort of shared risk model, then the threshold for acceptable side effects for male contraceptives would be similar to that for female contraceptives. So there's been some work in ethics about this and some papers published, and we're hopeful that regulatory agencies will consider this as a risk model in the assessment of male contraceptives as they move forward in development. And then lastly, just as a reminder, drug development takes a long time. We're constantly asked how long, when, and I wish that the answer was shorter, but just as a reminder, this is just the timeline for the introduction of Plan B with ulipristal. This is a 30 to 40-year process. So we're hopeful that we can move a little faster, but these are difficult studies to do, and we need to do them for a long period of time in order to prove efficacy. So that is, again, one of the issues. And further, that these types of studies are very expensive. So looking for investors and people that can see that we have a very large potential market for these products will be critical in moving past the goal line. Okay, so just in summary, I hope that I've convinced you that unplanned pregnancy is a global epidemic and there's really an urgent need for expanded contraceptive options. We think that expanding options for men could potentially be a game changer in changing unplanned pregnancy rates. Reversible male hormonal contraceptives are in phase 2B and hopefully soon phase 3 clinical development. And there are novel non-hormonal male contraceptives that are currently in phase 1 trials, which is really exciting for the field. And really, to get these to the market, to get these across the globe to people that are interested and people who have unmet contraceptive needs, we do need regulatory guidance from the FDA, the European regulatory bodies, and so forth to really move these products forward. There has been some work, collaborative work, to try and provide guidelines to these agencies, and there's been some receptivity to them, but I think that's a major impairment to development because investors don't like uncertainty, and they would be much more likely to invest if the guidance was available as to what these products need to achieve. And then I just want to thank my collaborators, and if I haven't mentioned, and I hope I have, these studies that I've been privileged to be involved with are really team sports. We get funding from the NIH. We have collaborators at the Population Council. My close collaborator at the Lundquist Institute, Christina Wang, is the other PI of the Nesterone study. Regine Citrick-Ware at the Population Council and Diana Blythe are all involved. And these are all the sites that the Nesterone testosterone study was done at. And most importantly, I want to acknowledge the participants in these studies. Both men and women take risks when they're involved in clinical research, and we couldn't move science forward without them. So a big thank you to all the participants. And I'll be happy to answer questions.
Awesome. Well, I would just, if we could, at your desk, you can give a round of applause to Dr. Page for a lovely talk. And I just want to remind everyone that if you have questions, you can put them into that Q&A box. We do have one question in the chat that says, for unplanned pregnancies, which male age groups are principally responsible, and are those age groups well represented in the contraceptive trials?
It's a great question. So I don't know the data on unplanned, like who's mostly responsible. I would hypothesize that that will be men between the ages of 18 and 50. 50 might be a bit low. We in the hormonal contraceptive trials, that's the age group we've targeted that we include in our studies. And the reason we've used 50 is as the upper limit is that historically there has been some concern that testosterone could increase the risk of cardiovascular disease and prostate cancer. And if you look at, we don't, there's a lot of data that suggests that that's not the case when we use physiologic doses. And so, but we do know that the risk of prostate cancer has a little bit of an inflection point at 50. So to not do all that monitoring, which could result in telling people they have disease that they don't need to know about, has some risk associated as well. We've used 50. In the non-hormonal trials, I think they're going to try and increase that to 60 because that prostate and risk isn't, we don't think would be there. I think it's an important point. Men are fertile until death usually. And so the other reason I would say is that just, I think some of you may know this, but for cycles to be counted in contraception, the woman has to be 35 years or less when she has the cycle because that's considered her optimal fertility. So since we have that, we have to do that in terms of regulation. 50 hasn't been a huge problem, but I take the point that it would be important ultimately to test across the lifespan.
Great. I have a question about, as I'm sitting here thinking about this, just the idea of giving testosterone for men to take and thinking about all of the athletes out there and performance enhancing drugs. And is there any concern about overlap for those things? This may be a very naive question. Those doses, I also think about like the male unlimited, like go to this local clinic and they're gonna fix all your hormone problems. So where do those things overlap and what things are different in the contraceptive side versus, some of those other steroid
Yeah. So
sides?
that's a great question. So the testosterone dosing that we use in the Nesterone testosterone gel, for example, is very much keeping men in the normal range.
Mhm.
So the concerns about testosterone are really about excesses above the normal range. So we have been very diligent in dose selection. We actually started the study with a bit of a lower dose and increased it because we were seeing some men having low testosterone. So, in terms of steroid use per se, it is true. I guess if men wanted to, they could get 5 prescriptions for this and take a bath in it. But it's not really, there's not enough there to be concerned about that. It's really just replacing their natural production of testosterone.
Mhm.
Now, in terms of sports and if men were using this and drug testing and all that kind of stuff, well, it would be good to have to deal with that problem as far as I'm concerned, and not and stopping a drug development for contraception because we're worried about the impact on professional sports seems misguided to me.
Sure.
But it is a point. I mean, testosterone is a controlled substance, but you know, we don't see women, estrogen's not,
Mhm.
but at this point anyway, we don't see estrogen abuse from contraceptives.
Sure. Sure.
So it's an important point, but I think if we can stick with physiologic dosing of testosterone,
Mhm.
we'll just work through the regulatory issues as they arise.
Yeah. There's a question, a logistical kind of question. You said the gel has an alcohol base. That's not been a problem for acceptance, is the question?
No, because so it's put on the skin. So it's just when I say that, I mean that just like hand sanitizer, that's an alcohol-based gel. And what the alcohol base does, I mean, it just means there's alcohol in the stuff. And from your use of hand sanitizer, when you put it on, it dries really fast. And that's the same thing here. So it also helps with the absorption. So it's just a part of the gel per se. And it actually also, because it dries quickly, that helps with lack of transfer. People do need to wait 4 hours to shower after they use it to maximize the absorption and minimize prolonged skin-to-skin contact with another person or a child. So there are some, but that's the same as for testosterone gel that's used for testosterone replacement.
Mhm.
So yeah, the alcohol is just kind of, some people like the smell, some people don't.
Um, there's a question about what future changes to research models look most promising. So that's the first part of the question. And then what is a realistic improvement regarding expediting male contraceptives and reducing time to develop and time to market?
Yeah. So I think, even though I work in hormonal contraception, I will say that I think it's really exciting. The most exciting thing that's happening now is that there's finally movement to get some of these non-hormonal products to clinical trials. And I think that's coming with, in answer to your question, it's a very collaborative field. And there's been a real movement to get experts in all these different development and companies and everything to try and come together and say, what should the guidance be for development? Are there surrogate markers we could use? What can we, how do we get things to evaluate them in pregnancy? Because there are some gaps in knowledge that are making that process more difficult. So I think the most exciting thing is actually seeing the field come together to try and provide these agencies with some guidance. So that I think has been great. What was the second part of the question?
Realistic improvement regarding
Hmm.
expediting male
Yep.
contraceptives, time to development, time to market.
Yeah, so I think that the biggest, my anticipate that if we can get one product to the market, whether it's hormonal, non-hormonal, or whatever. Um, Even if there isn't like huge uptake of that product, that will really set the stage because once people have experience, even if it's not personal experience, they're seeing a friend, they've heard about it, it changes the way people think about the whole concept. So I think the way that we will accelerate is what we need to do a phase 3 study of any method is funding, be that public, private, NGOs, whatever. Um, and then I think that getting something out there will really pave the way and I think investors then will see, yeah, people want this. People want choices. There's a lot of men who want contraception.
Yeah. Um, there's a question that starts with great talk. For
Thank you.
unplanned pregnancies, do you feel that the development of newer male contraceptive options would fuel or evolve sex education to a point that would also benefit family planning for the young adults? So those 18 to 26-year-olds.
Yes, I really do. So there's great data. There's like data from Malawi, there's data from all over Africa that supports the notion that male engagement or male education about contraception, even though the contraceptives are mostly female, increases uptake and use. And there's also, the flip side of this whole thing is that young men don't go to the doctor. And there is really a lack of male engagement in healthcare until 50 because men don't need a pap smear. They don't need contraception, like they're just doing their thing. And so we actually think these products could help to get men engaged in preventative healthcare for a variety of things, not just contraception. And even though that might be an initial investment, it could have downstream positive economic impacts for healthcare. So, yes, I think that education about contraception for men and normalizing these things will be effective for both female and increasing female and male method uptake. And I think these could be ways of actually improving male health in the long run. Hypertension, like who knows if they have hypertension if they've never had their blood pressure checked, right? So,
Right. Yeah. Yeah, and I think it's so interesting how reproduction can be such a great marker for your other parts of the health of your body, right?
yep.
And so yeah, that's an excellent point. And the person that asked that question also felt that that was an excellent point. So
Thank you.
um awesome, awesome. I don't see any other questions. I'm just gonna give you guys like one or two more moments here. I think my last question So if anyone has a quick one before I finish this. But my last question is just, I'm a very basic researcher. I do things that aren't anywhere near the marketplace. So how do we work on that continuum from those of us doing really basic research to interface with, people like you who are doing things that are closer to being used by individuals. And yeah, how do we continue to bring those ideas to you?
That's a great question. And I think obviously one of them is having interfaces like this. I think, we used to get some of that at meetings, at conferences and so forth. That's become very specialized too. You go to different conferences than I go to, right? So
Hmm.
that interplay has also, and we don't do as many things in person anymore. So I do think funders have a role in trying to develop funding opportunities that require that kind of interface. I think it's a really tough problem, though. I mean, it's also a problem for trainees because they
Hmm.
aren't getting the exposure to either end of the spectrum if they're in
Hmm.
one place. And we're, currently seeing a big problem with the pipeline for a variety of reasons. So I think it's a really, I don't think I have an answer.
Sure.
But I do think there is a role for, especially people or funders like Gates, and I wish the NIH, to recognize that problem and the siloing that's going on in science,
Hmm.
and trying to create these interface points because we're all just like head down sometimes.
Yeah.
I think, you could be developing something that I think could make a jump and we don't even talk to each other. So I don't have a great answer, Jennifer. I'm
Yeah.
sorry.
No,
If I
no,
think
that's
of one,
fine.
we can write a grant about it. How about that?
Absolutely. That would be fabulous. You're getting lots of thank yous. There is one plug about building some of these opportunities through the Contraceptive Technology Innovation Exchange, which I'm not familiar with, but if you go to, yeah, I'll
Okay.
grab this comment and
Great.
maybe we can stick it onto our uh, SSR website. So if people wanted to check it out, they could do that. So
Yes, please. And if you have questions, I'll provide my email and can certainly willing to reach out. I guess
great.
my last take home is be positive. Male contraception can come. We can make a difference. So, so I look forward to having more positive things to talk about in the future.
Great, great. Thank you so much.
Thanks, everyone.
And thanks to everyone who joined us.